Hello everyone, Mrs. Hoadley here again, and this is the second of two PowerPoints over genetics. This one's going to cover complex patterns of inheritance. And I just want to remind you that I also have notes posted on Google Classroom, detailed notes covering the PowerPoint with many examples, and the examples are going to have answers. The answers are always going to be found in red. So please look at that as well. So the previous PowerPoint talked about Mendel's law of heredity, and we're just going to review that one more time. Mendel came up with some laws or some principles. His first one was the principle of dominance, and we looked at that in the previous PowerPoint. Now, there's always going to be two forms of each gene, and those are going to be known as the alleles. The dominant allele will always be expressed over the recessive allele. Remember, the dominant allele is going to be represented by uppercase or the big letters. The law of segregation. Now, this describes the behavior of chromosomes during meiosis when the homologous chromosomes are separated. Now, remember homologous chromosomes. Think of that word homo. Homo means the same, okay? Now, homologous chromosomes are going to be the same size, the same shape. They're going to have the centromeres in the same location, the same genes as well, located in the same spot. Homologous chromosomes. This law states that the two alleles for a trait separate or segregate when the gametes are formed. Number 3, the law of independent assortment says alleles will randomly sort into gametes without interference from other genes. Okay. Now, the previous PowerPoint had examples of monohybrid crosses where you were looking at complete dominance. And that's when one allele is completely dominant over another. For example, if we take two plants and they're going to be crossed for the height of, for the trait of height. Let's say we're going to cross one parent plant that's tall and one parent plant that's short. Okay. Now, the P generation just refers to the parents. So one parent is going to be homozygous, two big T's. The other parent is also going to be homozygous, but two little t's. Now, the F1 generation refers to the first generation or the generation of the offspring produced by the cross of the parents. And in this case, the monohybrid cross that we see over on the left-hand side is going to produce the children of the offspring that are going to be all heterozygous. Okay, heterozygous, tall. If we look at coming up with a genotypic ratio, remember the genotype, I can't see your genotype. I can't see the genotype of this plant. I can see the phenotype. Remember, phenotype is the physical expression, what we can physically see. But if I look at the genotypic ratio, I have no offspring plants that are going to have two big T's. I will have 4 that will have big T, little t, and none that will have two small t's. And they're all going to appear to me as being tall. Okay, so we could say they are 100% tall, or if we're looking at a ratio, we would say 4 tall to none that are short. Now, this PowerPoint will cover complex patterns of heredity. We're going to look at incomplete dominance, codominance, multiple alleles, and sex-linked traits. Okay. Now, the first one, incomplete dominance. Well, for many, many years, scientists had realized that not everything we inherit follows Mendel's rule, his principle of dominance. Some things are inherited differently. One type of inheritance, the incomplete dominance, neither allele is completely dominant. So the heterozygous individual or the heterozygous offspring is going to be a blending or an intermediate of the two traits. How I like to remember it is the IN in incomplete is the same as the IN in intermediate, or another way of looking at it as a trait that's going to be in between. Okay. One parent, in this case, it's a red flower. Okay, and the alleles for the red flower are two big R's, crossed with another flower, a white flower, and the alleles for the white flower are two big W's. When you do your monohybrid cross, when you cross the red and the white flower, all of the offspring flowers are going to have the alleles of one big R and one big W. Okay. So the resulting offspring are going to be a color that is going to be a blending or an intermediate between the red and the white. So that's how we produce the pink offspring. In humans, a classic example of incomplete dominance would be the type of hair that a child could inherit. So for example, if one parent has straight hair and the other parent has curly hair, often what will happen is the child will have a type of hair that is intermediate or in between the straight and the curly, and that would be wavy. The next pattern of inheritance, complex pattern of inheritance, is codominance. With codominance, both alleles are dominant, so both are expressed at the same time. So for example, with codominance, co, guys, means together. If we had a white and a red flower and we crossed them and we were looking at the pattern of type of inheritance called codominance, you're going to produce offspring flowers with both red petals and white petals. Okay, both colors are going to be shown together, together in the offspring. Okay, now another example of codominance is found in roosters. So we're going to have a situation where we're going to cross a black rooster with a white rooster. Okay, so we have codominant example here. Okay, the black rooster's alleles are going to be two big B's, and the alleles for the white rooster are going to be two big W's. Okay, now we're going to cross these. And remember, we're talking about codominance, together. So when we cross the two big B's and the two big W's, we are going to have all of the offspring roosters are going to have a genotype which will be they're going to inherit one big W and one big B. Okay. Now, because it's codominance, this offspring rooster is going to appear. It's known as a speckled rooster because you're going to see both colors in the offspring. So the speckled rooster is going to have some white feathers from one parent and some black feathers from the other. Okay. Now, to define it, we'd say codominance is when we have two dominant alleles that are expressed at the same time or together, meeting together. Here's another example. If you take a red horse and you cross it with a white horse, you're going to end up with an offspring horse that's going to have both red and white hairs together. Something I want to explain about this. Okay, now I could write the alleles for the red horse as two big R's. I could write the alleles for the white horse as two big W's. And if we cross it, we do a monohybrid cross, the offspring horse that appears with both colored hairs in it, I could also write it as big R, big W. Sometimes you'll see it written like this, where the alleles are actually going to be known as superscript, and the C right here just represents the gene. Okay, the gene for color, and the different alleles are written up here as superscript. Okay, so you might see it both ways. It still means the same. Here's an example where we're going to cross a red bull. Okay, so right there is the gene for color, and the alleles are up here, they're superscript. But again, guys, I could write it as two big R's, and then cross with a white cow. Okay, I could write it like this. Now, the offspring cow is going to be known as a roan colored cow. So there's the genotype for the red bull. There's the genotype for the white cow. And then all the offspring are going to be represented as big R, big W. Okay, so I could write it like this. Either way works. Okay, now this is a special color they call cows when they have a combination of both white and red hairs and offspring. So this is the bull crossed with the cow, and this is the offspring right here. Okay, the roan colored offspring. Okay, now another example of codominance is sickle cell disease, and some of you guys may have chosen this as your genetic disorder that you're going to research for the QPA. But let's take a look at a little look at sickle cell disease, or sometimes called sickle cell disorder, and it's another example of codominance. So it's a disease in which the hemoglobin, and that's the molecule in the red blood cells that carries around the oxygen, it's malformed. Red blood cells are usually nice and circular, but if somebody has sickle cell disease, the red blood cells are called a sickle shape that represents sort of a half moon shape. The sickle shape results in, just because of the shape of it, it results in slowing down blood flow and will actually even block some smaller blood vessels, so the blood can't get to the tissues of the body. This then results in damage to the tissues from the lack of oxygen, and it can cause extreme pain. The only treatment we have for it right now is the person, if they're having what's known as a sickle cell crisis, they can receive very powerful pain medication, and then they'll often have blood transfusions. Right now, there's lots of research going on in the area of gene therapy. Again, that would be classified as genetic engineering, to see if they can replace that faulty gene that causes this particular disorder. So if we look at red blood cells, this is what they normally should look like. They should be nice and circular. And this is an example over here of somebody who has sickle cell anemia or sickle cell disease. These are these sickled shaped blood cells. So it's an example of a codominant trait. And so here, the big N represents the gene for red blood cells. And superscript, the allele up here, the big A represents somebody who has normal red blood cells that have those nice circular shaped. Somebody who has a sickle cell red blood cell would have the allele big S that would produce these sickled crescent-shaped red blood cells instead of the circular ones. So if we look at a cross between one parent who is presenting with normal red blood cells and another parent who carries the allele for normal red blood cells, but they also carry the allele for sickle cell. So when we cross and we get the 4 possible genotypes of the 4 possible kiddos, we are going to have, so when we cross them, we're going to have one child presenting with this genotype, the second child presenting with this genotype. We're going to cross here with that resulting genotype, and here for this one. So in this situation, we are going to have 2 of the 4 kiddos, these 2 down here, are going to present with normal red blood cells. The other 50%, or the other 2 kids up here, we're going to say that because they have the allele right here, the big A, they're going to appear as not having the disorder, but because they have or they carry the sickle cell allele, they are going to be known as carriers. So potentially they could pass this disorder on to their future children. Okay, so a carrier carries that allele, but they themselves do not show with the disease. Okay, because remember, if you were going to have sickle cell, you would have to have you'd have to present with two big S's. Okay, so an individual with the two A's will make normal blood cells. An individual who presents with a big S and a big A, okay, are going to make some normal blood cells, okay, because they have the big A, and that they're going to produce a few sickle cell blood cells. But we're going to say that they are the carrier, okay, for that sickle cell allele, because they can pass on that trait. And then an individual that presents with two big S's will only make sickle cell shaped blood cells, and they will be the individual who will have sickle cell disease, also known as sickle cell anemia. Okay. Now, human blood, that is also an example of codominance, but it's also an example of another pattern of a complex pattern of inheritance called multiple alleles. Now, blood type is determined by the presence of an antigen, and an antigen is just a small little carbohydrate on the surface of the red blood cells. So a cell can have an A antigen on its surface, and that would then make that person have what is known as type A blood. A B antigen on blood cells would then indicate that that person has type B blood. Both antigens on the surface of the red blood cell makes the person have type AB blood. And if neither antigen or no antigens are present on the surface of the red blood cell, then that person has type O blood. Okay. Now, looking at the chart over on the right, guys, phenotype. Remember, phenotype is what we can physically see. Well, blood, I can't physically see the difference between A blood, B blood, AB blood, or O blood. Blood just appears as blood. So when we're talking about phenotype, we will talk about type A blood, type B blood, type AB, and type O. Now, we're going to have these things, the genotypes, and we are going to have a couple of different genotypes for type A and type B blood. Okay. Now, if I go over here onto the left-hand side, blood is going to have three different types of alleles. We're going to have, and so blood is going to be represented by the gene for blood with the big I. Okay, and we can have three different type of alleles. We can have A, B, and then a lowercase I. Okay, and the lowercase I just says that it's recessive. Okay. Now, we say that blood is codominant because the big I A and the big I B are codominant over the recessive allele. So over here, guys, we can say that if you have type A blood, you may be homozygous for type A blood because you're going to have both alleles being big A. You may be heterozygous for type A blood. Remember, heterozygous means different. So you're going to have the big A and the recessive allele right there. Same thing with B blood. You can be homozygous or you can be heterozygous. Homo means the same. Hetero means different. If you have type AB blood, you're just going to appear with both alleles, the A and the B. And if you have type O blood, then you're going to have two small recessive alleles. Now, just want to show you something here. I'm just going to click through until I get the chart here. So again, blood has three types of alleles: big IA, big IB, little I. And then big IA and big IB are going to be co-dominant over I. I is recessive. So remember, my phenotypes are my types of blood, A, B, ABO. These are my possible genotypes. Sometimes we will write the genotypes a little bit different because this is biology and we have to make everything really confusing. So sometimes we'll write the genotype for type A blood. If you're homozygous for type A blood, is two big A's. If you're heterozygous for type A blood, sometimes we'll write that as a big A and an O. Okay. Same thing with type B blood. If you're homozygous, sometimes we'll write it as two big B's. And I have a couple of practice problems for you guys, and I want you to just use the two big B's, the two big A's, and so forth. If you're heterozygous for type B blood, you're going to be B and O. AB blood is only written one way, AB. And the two little recessive I's are represented by an O. Okay, so remember, type AB blood is representing codominance because they're codominant over the recessive little I. The universal recipient is known as type AB blood, and they can receive blood from, because remember, they're going to have an antigen on the surface that will indicate that it's A blood, and an antigen on the surface that will indicate that it's B as well. So that just means because of both the A and the B antigens on the surface of the blood, they can receive blood from somebody who is type A, who has the A antigen on the surface, somebody that has type B blood that has the antigen on the surface, of course, somebody who has AB blood, or somebody has type O because O has no antigens on the surface. But because they have both, type AB blood has both A and B antigens on the surface, AB blood can only be donated to another person who has AB blood. So this is why, well, in terms of blood, like A is most common, O is the least common. O, it has no antigens on the surface, and because it has no antigens on the surface, it can only receive blood from other people who have type O blood, but it can be put into any type of person. So somebody who has type A, B, AB, or O can receive type O blood. I'm not going to ever ask you questions about universal recipients and universal donors and so forth. I just out of interest and knowledge moving forward, especially when you become of age and you think that you may want to donate blood one day. You'll kind of have an idea of who you can give your blood to. Okay, now sex-linked traits. This is the next complex pattern of inheritance. These are traits that are going to be located on sex chromosomes. For our sake here in sophomore biology, we're just going to talk about traits that are going to be carried on the X chromosome. But remember, X and Y are my sex chromosomes. Some classic examples of sex-linked traits are hemophilia, and hemophilia is a disorder where an individual has a difficult time clotting their blood. Another one is muscular dystrophy, where a person is born with muscles attached to their skeleton, for example, and these muscles will slowly start to waste away, and the person does not have the ability to build muscle mass or replace the lost muscle. So, sex linkage. If a trait is autosomal, okay, remember autosomes are the body cells, it's going to appear in both sexes equally. If a trait is sex-linked, it's usually going to be seen only in males. Genes that are carried on the X chromosomes are called sex-linked genes because they're inherited with the chromosome that determines gender. So most sex-linked traits, though, that we're going to look at are going to be recessive. Now, traits that are determined by sex-linked genes are called sex-linked traits and can be predicted with Punnett squares using the X and the Y. So when you're trying to figure out Punnett squares for sex-linked disorders, not only are you going to determine what alleles the offspring are going to inherit, but you also have to account for the sex as well. So remember, a female is a female because she has two X's. For her to express a recessive phenotype or recessive sex-linked disorder, she must have two recessive alleles. For the male, remember males are X and Y, he needs to receive only one recessive allele to express that recessive disorder, okay, the phenotype. And this is the reason why more males are affected by sex-linked disorder. Okay, so take for example color blindness. I'm going to remember if it's sex-linked disorders, I have to account for the X and the Y chromosomes. So on the X chromosome, if for color blindness, if you have a big C, okay, as the allele, that's the normal allele. If on the X chromosome you have a recessive form of the allele, the little c, then on that X chromosome you're going to carry the allele for color blindness. And then Y, this is a normal Y chromosome. And for simplicity's sake, guys, as sophomore biology students, we are only going to talk about sex-linked traits carried on the X chromosome. There are a few sex-linked traits carried on the Y chromosome, but we don't get into that at our level of biology. So we're going to have, remember we're talking about color blindness. So if we have two big C's, and remember it's two X's make a female or a woman, she's going to have normal vision. If the woman has a big C allele and a little c, then she's going to appear as having normal vision, but she's going to be a carrier for the color blindness allele. If a female has two small C's, she's going to be colorblind. If a male, because remember X and Y makes a male or a man, remember we're gonna assume at our level of biology that there's nothing carried on the Y chromosome. If the guy has a big C on the X chromosome, he's going to have normal vision. But if he inherits the small c, then he's going to be colorblind. So remember, a carrier is somebody who's going to be heterozygous for the recessive trait. Okay, so looking back here, colorblind woman has to receive or has to have the small c, the recessive allele on both the X chromosomes. Where a guy, for him to be colorblind, he only has to receive one copy of the recessive allele. So this is the reason why there's more guys that will have color blindness than women. So in example, guys, what we're going to work through right here is going to be red-green color blindness. And remember, the allele for color blindness is going to be carried on the X chromosome. So what we're going to do is we're going to have mama here. Okay, two X's makes a female. She's going to be a carrier for the colorblind allele. She's going to have big C and little c. Now, Dad over here, he's got normal vision because he has on the X chromosome a big C. So when we figure out sex-linked inheritance, guys, we have to, first of all, and what I do to make it a little bit easier, I always cross the sex chromosomes first. So the first thing I do in the boxes is, like, for example, I would cross big X and big X, put that in the box. So we're going to have a female. Cross big X, here's another female. Cross big X and Y, we've got a male. Big X and Y, we've got another male. And then I go back and then I put those alleles that are superscript next to the sex chromosome. So I'm going to put the big C here, the big C here. I'm going to put the little c and the big C, and then the big C and the little c. So you have to account for this type of inheritance. You have to account for the sex of the offspring, and then also the inheritance pattern of the alleles. So in this case right here, this girl is going to be normal. This girl is also going to be normal. Mind you, she's a carrier for that recessive allele that can cause color blindness, but she herself is not going to be colorblind. Okay, kind of think of it as the principle of dominance. As long as we have the big C, that's going to cover up and mask the little c. So she's going to be normal vision. Okay, this guy down here, normal vision, but unfortunately this guy down here, he's going to be colorblind. Okay, so I want you to think that the mother is actually the individual who's going to determine if the son is going to be colorblind or not. Because remember, dad determines the sex. He's going to give the Y chromosome, and mama is going to give the recessive allele on this X chromosome that causes that son right there to be colorblind. Okay. The last thing we're going to look at is something called pedigrees. And scientists can look at a family history of a genetic trait or disease to determine how it's inherited over several generations. So this pattern of inheritance can be mapped on a chart, and the chart's called a pedigree. Pedigrees can show the phenotypes, and also you can use the pedigree to determine possible genotypes. So in a pedigree, an individual whose genes have a recessive trait but express the dominant trait, remember that's called a carrier. Okay, because they have that dominant allele, that is going to cover up and mask the recessive allele. But remember, they are still carrying and can pass on that recessive allele to their offspring. That's why they're known as a carrier. So when you're looking at a pedigree, and just to let you guys know that I have examples in the notes that are attached in Google Classroom with questions, and then the answers are typed up in red. So it's a way to kind of follow through with some pedigree examples. So with a pedigree, squares are going to represent males. Females are represented by circles. That if there's a horizontal line, that's going to indicate marriage. A vertical line coming down would indicate the offspring or the children. Often what will happen is you'll see half of a circle, or you can also see half of a square being colored in. And then that would just represent that the male or the female are a carrier. Okay, so again, females are represented by circles, and the square is going to be representing a male. Okay, a line between a horizontal line indicates marriage. A vertical line indicates children. Now this couple right here, they had 2 kids. Now the birth order is always going to go from, you're gonna be able to see it. Birth order is going to go, firstborn is always going to be on the left, and then the last born. is going to be on the right. So in this case, we would say they had two kids. The first one was a girl, the second one was a boy. Okay, now, so if someone in the family had a certain disorder, such as color blindness, the circle or the square will be colored. Okay, half colored like this. So often when the circle or the square is completely colored in, that just means they have the disorder, and if it's partially colored in, then they're carrying the gene for the disorder. So this part right here is going to be really important because this will help give you an idea of how a particular trait is inherited through many generations in a family. So if it's an autosomal trait, okay, so remember that's going to be a trait on one of the body cells, or body chromosomes, or the non-sex chromosomes. It's going to appear equally in both sexes. So we can have equal distribution of the trait among the males and females in the pedigree. If it's sex-linked, trait whose allele is located on the X chromosome, it'll be seen mostly in males. So in order for a female to inherit a sex-linked trait, then she must have that inherited trait appearing on both of the X chromosomes, and then only on one of the X chromosomes for the male. Okay. Females that inherit the allele from one parent, from only one parent, then they'll be known as being a carrier. A dominant trait, well, every individual with the trait will have a parent. They have to have a parent that has that trait. If it's a recessive trait, and usually dominant traits, you're going to see it appearing usually in every single generation. A recessive trait, every individual with a recessive trait, and this is the tricky part, they can have one, they can have two, or they can have neither parent exhibiting the trait. So for neither parent exhibiting the trait, that just means both parents are heterozygous or carriers for the trait. And often with recessive traits, they're going to skip. They may skip a generation as well. You may not see them in every generation. Okay, so here's an example of a pedigree of a dominant genetic disease. Okay, mother doesn't have the trait. Dad does. It's colored in. Couple 1 and 2 had 2 children. They had a girl first, and then they had a boy. Okay. That this girl married a guy, and they had 1 kiddo that had the trait. This son over here married this girl, and they had 1 child who doesn't have the trait. The numbers over here, the 1, the 2, and the 3 represent the 3 different generations. So first generation are the parents, second generation are their children, and the third generation refers to the grandchildren. Okay, so dad here has the trait, daughter has the trait, son has the trait. Okay, so you can see in this case, it's a dominant genetic disease because it's appearing in every generation, and you have to have at least, to get that dominant genetic disease, you have to have at least 1 parent having the disease. Okay, so that's a dominant genetic disease. The next one's a recessive genetic disease. Okay, mom and dad again had 2 children, birth order, girl was born first, and then the boy. Okay, this guy is through marriage, this girl is through marriage. She has the recessive genetic disorder, and so does she. Okay, now I can see that it is a recessive genetic disease because often it's not going to appear in every generation. And remember, with the recessive trait, it's kind of, with this is kind of a little tricky because remember, you can have 1, both, or no parents show the trait, even if the children do. Again, there's 3 generations of folks in this pedigree. This is an example of a recessive genetic disease. The last one is a sex-linked genetic disease. He doesn't have it. She, right here, the mother, it's kind of hard to see, but it's supposed to be half shaded in. That just indicates that she's a carrier. They produce 2 children, birth order, first a girl, then a boy. He has a particular disorder, and then the daughter down here, okay, she's a carrier. So remember, with sex-linked traits, you're going to see most often these traits are going to appear in males because males only have to receive 1 copy of the recessive allele on that X chromosome in order to receive or get the disease. Remember, dad determines the trait. So for this kiddo right here, because he's male, dad can only give a Y chromosome. And mother, because she is a carrier, so she has a big C and a little c. This is the X chromosome that she passed on to her son. So mother is responsible for passing on sex-linked recessive disorders.